Welcome to Mr. Chalk's Revision Tips. In this video, we will look at light microscopes, magnification, and electron microscopes. So, a light microscope is an instrument that uses visible light and magnifying lenses in order to go and let us see small objects that we couldn't necessarily see with the naked eye. So the usefulness of any microscope is that it produces a better resolution than the eye. So when a light microscope is it's important to start off with a low power objective lens, as the field of view will be wider. Right, so this increases the number of cells that you'll be able to see. Right, this makes it easier to find what we are looking for. Then ensuring the cells are in the middle of the field of view, rotate to a high power lens, and then you can start to see it at a high magnification and in more detail. So if we look at this, so suggest why ribosomes cannot be seen with a light microscope, it's because they're too small, or because a light microscope does not have a high enough resolution or magnification. And when viewing a specimen for the first time, which objective lens should you use? Well, it's the lowest magnification first, because it gives the widest field of view. So magnification, like we've said, is a process of enlarging the apparent size, not the physical size of something. So this enlargement can be quantified with calculation, which is also called magnification. Right, so the way that we work out the magnification is that we divide the image size by the actual size. So if we go and look at this, so we know that the image length is 40 millimeters, and we know that we've got it at times 100 magnification. So if we actually wanted to work out what the physical size of the cell would be, you'd work it out by rearranging the equation to give you actual size would be image size divided by magnification. So that would be 40 divided by 100, which means that the actual size of the cell would be 0.4 millimeters. We look at this question. So the average diameter of a real blood cell is about 0.008 millimeters. So on the photograph, the diameter of the red blood cell is about 100 millimeters. So use the formula to calculate the magnification. So it would be 100 divided by 0.008, which would give you a magnification of 12,050. Look at the second question. So the figure to the side is highly magnified. The scale bar in the figure represents 0.1 millimeters. So if we're going to use a ruler to go and measure this, then calculate the magnification. So it's important to remember that you will be asked to go and physically measure things in exam questions. So the way that would work that out would be 40 divided by the 0.1 would give us a magnification of 400. When we look at using microscopes, quite often we'll want to be drawing cells. So biological drawing is supposed to be an accurate representation of what's actually observed, right? So it's not what you think should be there, it's not what you know should be there, it's not what you can remember should be there. So good biological drawing should be large with the same proportions of the observed cell. It should be labelled using separate ruled lines, so these should be spread out with a bullet point or clearly labelled, and make sure that the lines preferably go out, unlike in this diagram, horizontally from where you are labelling. So make sure that you give a title and make sure that it includes the magnification that you're using. You can go and also include in the title the size of the cell after you've worked that out. So electron microscopy is a technique for obtaining high resolution images of biological and non-biological specimens. It's used in biomedical research because it gives detailed pictures of what's actually in the cells, because it has a much higher resolution or magnification than a light microscope. So If we continue to increase the magnification past a certain point in a light microscope, the bits don't become visible, or you can't see them. So with an electron microscope, it increased the detail of what we can see because it's using the wavelengths of a beam of electrons rather than the wavelength of light. So most importantly, like we just said, it is at a much higher resolution. So the resolution can show details that are 0.001 micrometers apart. So this allows us to clearly see inside parts of the cell. So for instance, inside the chloroplast or inside the mitochondria. And electron microscopes pass beams of electrons through a specimen and have much higher resolution. But because you're passing a beam of electrons, the specimen has to be dead, so we can't look at live specimens, and it has to be done in a vacuum. So if we're comparing the advantages and disadvantages of light and electron microscopes, some of the key things that you might want to be put in are that electron microscopes have a high magnification, they have a high resolution. Scanning electron microscopes can be used to examine the surface structure of cells. However, light microscopes can be used to examine living cells, whereas electron microscopes have to look at dead cells.
Thanks for watching.